Okay, so I would like to welcome everyone back to our special session this morning, and also we have an online audience. So welcome everyone who is watching online, and I want to encourage you folks to ask questions. There's a chat box there, so feel free to ask your questions at any time during any of these three talks. For those of you who don't know, we do have an audience in the room here. This is part of our advisory council meeting, so we do have a live audience in the room for these speakers, so we'll hear from them as well. But I want to get started right away to give the speakers enough time to talk about some very, I think, important and critical issues with some pests that most of us are familiar with, whether intimately or not. But our first speaker is Paul Leisnham, and he's an associate professor in the Department of Environmental Science and Technology. And he specializes in mosquito ecology and management and watershed health and sustainability. So Paul has a PhD in ecology and health from the University of Otago, New Zealand, and a BS and MS in zoology also from the University of Otago. So Paul, welcome, and please share with us what you have.
Okay, well, thanks very much. Well, it's a real pleasure to be here today. Thanks for inviting me to speak. I think I spoke at a similar event 3 years ago and really enjoyed myself. And then I spoke a little bit about some of the social science work that we've been doing on mosquitoes in Baltimore, particularly looking at how knowledge and attitudes affected resident-based practices, the effectiveness of education outreach. Today I'm going to focus more on the ecology of mosquitoes in Baltimore City, but in particular how that ecology intersects with social issues at different scales. So hopefully it's going to be of interest to you. My background is what was mentioned, is in ecology, and I started studying mosquitoes to really test some basic ecological theory. They're a great model system, particularly these species here that utilize water-holding container habitats. But of course, mosquitoes present a threat as a pest and as a vector of disease, and their management is particularly difficult. So over the last 5–10 years, I've really focused on social and ecological issues affecting their management. Okay. Great. Okay, before I start, I'm going to recognize a number of collaborators that are working with me in Baltimore City, in particular Shannon Ledoux from the Carey Institute of Ecosystem Studies in New York, as well as Rebecca Jordan, Dawn Beeler, and Jacoby Wilson, and a number of community partners and connections, most notably Parks and People Foundation here in Baltimore City. So these people have been instrumental in gathering social and ecological data, and a lot of this has been driven by, of course, graduate and undergraduate students. I'll present the work that is mainly being synthesized by a graduate student, Eliza Little, who's working closely with Shannon Ledoux, and one of my undergraduate students, Chris Austin, in just a moment. And of course, we're funded by the Northeastern Integrated Pest Management Center, and we were able to leverage the work out of that grant to secure an NSF, Coupled Natural Human Systems program. That's been driving much of our work for the last 3 to 4 years in Baltimore City. Okay, so much of our focus has been looking at this two-way feedback between social systems and pests, particularly container utilizing mosquitoes, where urban decay leads to greater mosquito habitat, mainly through trash and adult exposure. This increased abundances of adult mosquitoes leads to reduced use and valuation of the outdoor environment. This is kind of the conceptual framework that we place our work. And so today I'm not going to address all of that, but some little snapshots out of that. I'll start my talk with a very brief background on urban mosquitoes and disease risk. Many of you are probably very familiar with this. And then I'll address a synthesis of abundance data from Baltimore, Maryland, that provide some interesting insights on how mosquito populations may be regulated in the city. And then I'll also finish with a more specific study looking at the effects of container condition on competition between the principal pest in Baltimore City, Aedes albopictus, the Asian tiger mosquito, again a species which many of you are probably very familiar with, and the northern house mosquito, Culex pipiens, which is also invasive, but it's been here for over 400 years. It's much more of a resident mosquito. Okay, so for mosquito-borne disease, risk is really related to how likely a mosquito is to pick up a new infection and pass it on, right? How likely is an adult, female adult, going to bite you, infect, and pass it on? And it's also a function of density of adults, their survival or longevity, and biting behavior. Of course, they may not just solely bite humans, but they may bite a whole range of other vertebrates. And we look at these components, of course, in this model of vectorial capacity, which looks at infective bites per person per day. And again, many of you will be very familiar with this. It has density estimates, adult density, biting rates, looks at daily adult female survival or longevity, as well as vector competence and extrinsic incubation period. These last two components are often very specific to the vector host pathogen system, but these first three are quite robust and generalizable across a number of different systems. And changes in density, biting rate, and survival can influence risk in pretty predictable ways. Here I'm just showing a schematic of how vectorial capacity may change when we double density, biting rate, and survival. So each little bar reflects a twofold increase in vectorial capacity. And the relationship between these components here is really a linear to exponential relationship. So if we double the rates of all of them, we get much higher vectorial capacity. If we double the rates of density and biting rate, but we halve survival, we get slightly less vectorial capacity. So these sorts of ideas were kind of encapsulated in this review paper that Shannon Ledoux led, on which I co-authored with Brian Allen, Michael Levy. And it mainly looked at environmental conditions that may influence these sorts of factors and, in turn, vectorial capacity. And of course, control and management has historically focused on reducing individual mosquito survival. And we've historically done that by applying larvicides. Right? We get a lot of bang for our buck with reducing adult female longevity. Really reduces vectorial capacity. But we're in this post-DDT era, right? We're aware of the environmental and human health concerns with many adulticides, and so our focus has shifted to reducing densities. And that often occurs at the larval stage. Right? And that can occur applying more biorational larvicides that have much fewer non-target impacts, source reduction, reducing containers of water, and biological control. Although biological control is very difficult to do in container systems. And that's not to say that adulticiding isn't preferred or appropriate in certain occasions, but we're certainly shifting to these sorts of strategies. But controlling mosquitoes in urban landscapes is inherently troublesome, as I'm sure many of you realize. And container habitats are difficult to access and manage, right? They're numerous, they're often really cryptic, and they're on privately held land. So control agencies just can't access them easily and apply larvicides. So often larviciding is impractical. We rely upon residents to try and control these container sources. And so that's a really important tool in integrated mosquito management toolkit. And on the East Coast, we have a number of invasive species that are important vectors. Right? A lot of these species you'll be very familiar with. We've really captured some of the most prominent diasporas worldwide. Right? So Culex pipiens complex arrived in North America 400 years ago from Europe and Africa. Aedes aegypti, the yellow fever mosquito, arrived 200 years ago from Africa, principal vector of dengue around the world. And more recently, Aedes albopictus, the Asian tiger mosquito, arrived in the mid-1980s and used tires. That spread up and down the eastern seaboard. And Aedes japonicus arrived in the late 1990s, again in used tires. It's a very cold-tolerant species. It spread as far north as Canada, and it's spreading south as well. I think now it's in southern Georgia. So these species are here. Aedes albopictus, northern range, tends to peter out about southern Connecticut, New York City. Aedes japonicus is common in upstate New York. Aedes aegypti, of course, tends to be restricted to southern, eastern US. Doesn't extend much further than Georgia. And Culex pipiens is very common, or the complex is common, right up and down the eastern seaboard. So these are very widespread invasive vectors that utilize these water-holding containers. And of course, they're medically important. So the most prominent, as you know, the most prominent mosquito-borne disease in the US is West Nile virus. And here, these are human cases, reported West Nile cases in the Northeast region. Just some totals from the 12 states and District of Columbia that make up our region. We can see that West Nile has been a persistent problem. Okay, it peaked around 2003, but it's been a persistent problem. And in total, we've got over 2,000 total human cases. And of course, we know that West Nile is underdiagnosed, symptoms aren't obvious and clear. And it's vectored primarily by Culex pipiens, northern house mosquito, which circulates the virus and amongst bird populations that amplify it. But also Aedes albopictus is likely to be an important vector in certain situations, particularly urban situations, where it may bridge West Nile into human populations. And even Aedes japonicus has been shown, collected Aedes japonicus in the field has had West Nile virus, but it's probably a much, not really an important vector. Okay, and of course we're seeing the emergence of a number of other diseases worldwide and of course in the continental United States. And all of us probably very familiar with the Zika crisis. And when I took a look on the CDC website just last night, there's just over 1,500 total travel-associated cases in our region. And worldwide, the principal vector of Zika has been Aedes aegypti, the yellow fever mosquito. Right? That's been implicated as the principal vector. Of course, Aedes aegypti is not common in the Northeast region. But Aedes albopictus has also been shown to be probably a capable vector of Zika virus. And recent work is suggesting that it could drive an outbreak in this region. Right? This is a paper which was lead authored by Carrie Monroe from Tulane University. Shannon Ledoux, my collaborator, is a co-author on it. It's just using a simple epidemiological compartmental model to look at Zika and chikungunya outbreak, the potential in temperate cities in the US. They suggest that 1 out of every 2 infectious travelers arriving at peak mosquito season could initiate local transmission. That over 10% of introductions could generate a disease outbreak of at least 100 people. Right? The prevailing thought is that Zika is unlikely to outbreak in the Northeast because we don't have Aedes aegypti. And that we have Aedes albopictus, and it doesn't bite humans as often as Aedes aegypti. It bites a number of different vertebrates. But we could have an outbreak even if vectors feed from humans even just 40% of the time under ideal conditions. So this paper actually, interestingly, is still in peer review, but the journal releases it online, so you can go to the journal and look it up. And I can actually give you the link to this after the talk. So, and there is precedence for Aedes albopictus driving disease in a temperate city. 2014, Aedes albopictus drove an outbreak of dengue fever in Tokyo with over 180 people infected. So certainly there is a threat to our region from Zika virus. So I want to transition now to our work in Baltimore. And in fact, our study site is just through downtown, probably less than a mile from where we are today, in southwest Baltimore. And in particular, we're looking at mosquitoes across a socio-ecological gradient. And Baltimore is a really great city to do this because within a very small geographic range, you get a very steep gradient in socio-ecological and economic conditions. So we've got a study area with selected city blocks at the median household income, which is about 41,000, below that median and above that median. And the principal, by far and away the dominant species, are Aedes albopictus and Culex pipiens. And we see that among these neighborhoods, really the housing infrastructure and the green space is very similar, but the quality is very different. So these are just showing aerial photos of a high-income city block and a low-income city block. And just showing typical green space. Here we have yards, very well maintained. We have a number of containers that may be utilized by mosquitoes, like this planter. In comparison, in a low-income block, we see a lot of households that are vacant, right? So these yellow rectangles show vacant blocks, and in particular show houses where the roofs have collapsed. And in these areas, vacant lots, we often see piles of trash, a lot of trash. In the occupied lots, we often see very few functional or structural containers. Okay? Often they're just cement slabs in the back. So very different, but the housing structure and amount of green space is similar. This slide is just showing you a bit more about the difference. Here, again, we have a high-income block, very well maintained. And very close to it, we often see blocks with a lot of vacant housing, with a lot of trash. This is a tire dump here, which can produce larval habitat for mosquitoes. And in many blocks, we see vacant lots right side by side with occupied lots. Okay, vacant lot with overgrown vegetation. This is an occupied lot. We have occupied lots here in the background, vacant lots here in the foreground with a lot of trash, vice versa. Occupied lot here. This one does have a lot of functional containers. We can see trash cans, little buckets. These are functional containers. People use them on a daily basis. But in the background, with a lot of vacant lots. So a really interesting situation here in regards to urban mosquitoes. So one of our sub-questions of our work has been this one. Do trash containers in low socioeconomic status neighborhoods support greater Aedes albopictus production? Aedes albopictus is by far and away the most important pest mosquito. It aggressively bites people during the daytime, and we know through survey work in DC and Baltimore that people change their behaviors because of this mosquito. So in each of our neighborhoods, we have replicate city blocks. And over the last 4 years, we've done various interventions and we've collected a large amount of mosquito data. And I'm going to show you some of that data that's synthesized by Eliza Little with help of Shannon Ledoux and myself. So to give you a bit more of an idea, the data that I will present is from 2013 to 2015. We've also got 2016 data, which we're just processing at the moment. And essentially, we've collected adults at 24 sites over 12 focal blocks. We've collected eggs and larvae. We've done a lot of surveys to collect social science data. We've also done a number of focal groups. I won't talk so much about the survey data today. And for each of these years, we've really had three main sampling periods over the summertime: early, middle, and late summer. And the blocks kind of have this sort of structure where we have parcels, individual parcels, with an alleyway down the middle. Okay, so what do we find? Well, I'm going to show you data just from 1 year so that I don't confuse you too much. This is 2013. This year does show very general trends that are consistent across all of our years. And the main one is that high and medium income households, income blocks, this is looking at blocks, have very high Aedes albopictus abundances. This is adult female abundances. These abundances are much higher than high income blocks and well above typical threshold levels that mosquito control agencies use to trigger control. We don't see the same sort of pattern with Culex species. We really see no significant difference between their socioeconomic levels in adult female Culex. But I also want to draw you to one little aspect of these graphs, is that during this period in 2013, we saw quite low rainfall. And we saw quite different responses across socioeconomic level. We saw an increase in adult abundances in medium income blocks and a decrease in low income blocks. And we didn't really see much change in high income blocks. We also saw somewhat similar trends or a much larger increase with Culex. That kind of hints at how we think mosquito populations being regulated, which I'll summarize in just a moment. When we have a look at larval data, this is looking at larvae at the yard scale. We have yards with varying numbers of trash containers and then abundances here on the y-axis. We see that Aedes albopictus abundances are highly associated with trash containers. Right? Where you find trash, where you find yards with a lot of trash, you find a lot of Aedes albopictus. And we see more trash containers in low and medium income yards. We do not see similar trends for Culex pipiens. Right? We see no association between trash containers per yard and Culex pipiens abundances at the larval abundances at the yard scale. So what do we think is really driving mosquitoes across socioeconomic boundaries? Well, when we have a look at all 4 years of our data, we see some really interesting relationships. Firstly, we see that larval abundances at the block level is highly associated with rainfall, only in low-income blocks. We don't see that relationship in high-income blocks. And we think this is because people in high-income blocks are managing their water containers. This is consistent with work that was found in Washington, D.C. We see a pretty strong transition of larvae into pupae and pupae into adults in low-income blocks. We also see a similar transition, but a much shallower slope. Okay? So we see a much steeper slope here. And we think that that's, of course, due to management. Again, people are managing their containers, so we see much fewer larvae eclosing into adults. So we're really seeing two different forms of regulation between income categories. We're seeing environmental regulation in low-income blocks, a lot of rainfall. If you have high rainfall, a lot of mosquitoes, particularly in trash containers. Whereas in high-income blocks, we tend to see human regulation being much more important. When we look at median income blocks, we kind of see a mixture of both types of regulation. We don't see a strong association between rainfall and larvae, but we see a strong transition between larvae to pupae and pupae to adults. So we may see this kind of perfect storm where we have some yards with a lot of trash, and so when it rains, they produce a lot of Aedes albopictus. And then we also see some yards that do effectively manage. So what we've concluded is that human behavior plays an important role in defining spatial heterogeneity of Aedes albopictus. And weather conditions appear to interact with human behavior in different ways across socioeconomic boundaries. Aedes albopictus disproportionately utilizes trash habitats, and these are regulated by rainfall. And medium income blocks may support high albopictus under wet and dry rainfall conditions. So that was an interesting result. And I want to draw your attention to one little aspect that I alluded to just a few moments ago. And that was that we don't really see this trend in abundances with Culex species. Okay? We see similar levels of adult female Culex abundances across all socioeconomic status levels. And we also don't see, of course, this relationship between trash containers and larval abundances of Culex at the yard scale. And this is interesting. It's kind of consistent with work that we've done in Washington, D.C., where we do see a higher proportion of disused or trash containers and low socioeconomic status neighborhoods. And we see more functional and structural containers in medium and high income status neighborhoods. And we see that trash containers predict Aedes albopictus. We see that functional and structural containers predict Culex pipiens. And this trend could be the result of a couple of ecological processes. One, it could be that adult females are preferentially ovipositing their eggs in different container types. And we do know that Culex pipiens tends to prefer larger containers that are more permanent. It could also be because of inherent properties of the container that affect the outcome of competition between larvae. We know that containers, communities, mosquito container communities are structured by competition. Okay? These tend to be resource-limited systems, right? Detritus falls into them, and the mosquitoes browse and find particular organic matter and the associated microbes. And they're also largely ephemeral. Even functional and structural containers dry out eventually, so they lack vertebrate predators. And so competition is usually very strong. And we also know that Aedes albopictus is a really good competitor. Almost all lab and field studies show that it's competitively superior to all other resident species, including Culex pipiens. There's really been only 3 robust studies that have tested the outcome of competition between these two species, and they all show Aedes albopictus as being particularly competitive and strong. But little research has looked at resource conditions and densities typical of different types of urban containers. This may be really important because it could be structuring communities across socioeconomic boundaries. So there was this fundamental question: do functional and structural containers relax competitive impacts of albopictus on pipiens and promote Culex pipiens persistence? We would expect, with the competitive exclusion principle, that Culex pipiens should be driven to extinction. But we see it persistent, and it's an important disease vector of Western wildlife. So this was a particular question that was picked up by an undergraduate student in my lab, Chris Austin, shown here. And he developed two specific hypotheses. The first one was that interspecific competition is important in conditions typical to what these two species would find in field conditions. Right? We want to see whether or not competition is important. And the persistence of pipiens in some container types occurs because the contents alter the outcome of competition. Right? So there are the two hypotheses. And they really lead to testable predictions. Right? So we would expect that these two species would co-occur in field containers, and that the proportion of Culex pipiens would vary among different container types. Right? That's what we'd expect in this urban situation. And then competition among the larvae would be detectable at densities that we'd typically see in the field. And then competition would vary among aquatic conditions found in different container types. So three testable predictions from the two hypotheses. So what Chris did was he first looked at field data that we collected in 2015. And just as a reminder, we had sampled containers across our neighborhoods in early, middle, and late season. And during that year, we sampled 608 water-filled containers, and about half of them had mosquito larvae. And of those containers that had mosquito larvae, we were able to extrapolate an average density. Right? A number of containers didn't have mosquito larvae, we didn't include those, we just looked at those containers, if it did have mosquitoes in it, at what density were they found. This was really a baseline field density. And then Chris looked through our survey data and he identified the most common trash and functional containers. And amongst trash containers, plastic trash was really common. This could be all sorts of things like discarded planters, you name it, lots of things are made out of plastic. And also styrofoam trash, and he separated the two because styrofoam trash tended to be food containers that had been disposed. And also dump tires. And as a side, tires are a really interesting microcosm to study, particularly when it comes to mosquitoes. They can hold a lot of mosquitoes. And they're interesting in other reasons, which I won't go into. They also leach a lot of contaminants, which may differentially affect mosquitoes and may actually make them more tolerant to insecticides, which is another area that actually a student of mine's doing. So tires are just really interesting. It's something we need to take closer note of. Chris also identified functional and structural containers that were really common. These buckets, all sorts of buckets people have in their backyards, gardening buckets. They fill up with water, they add water to them, they hang around. Trash cans, right? The lids aren't put on properly, and they often fill up with water. There's various juices from food which seeps into them, very common in Baltimore. And then fence pole holes. So a lot of yards in Baltimore have wire fences. And when people no longer want these wire fences, they usually cut them at the level of the cement, and they leave nice little holes that fill up with water. So out of all these containers, Chris could calculate the proportion with mixed species, the proportion of total larvae that are Culex pipiens. Okay. So what then did Chris do? Well, then he wanted to do a competition trial. So earlier this year, he took samples, homogenized samples from containers in the field, and took them back to the lab. He divided the sample into 3 90 ml microcosms. After sieving them to remove all the field larvae and all the detritus. And then with those 3 microcosms, he applied one of 3 density treatments. to somewhat mimic Aedes albopictus invasion. There was a control treatment which had equal numbers of both species at the baseline field density. Then he had a reduced treatment, which was only Culex pipiens, and then he had an increased treatment, which had Culex pipiens and double the amount of Aedes albopictus. So all of the treatments had Culex pipiens, which is the inferior competitor, which is the invasive resident. It's been here for 400 years, and Aedes albopictus. Density was varied from 0 to equal densities to increased densities. And he applied this competition trial in the field. He had replicates for a total of 72 total microcosms. Then incubator set at typical summer conditions to isolate the effects from container contents. And he was able to look at the proportion survival and development time of Culex pipiens after 6 days, when larval competition is likely to be at its most intense. And he analyzed the data with standard linear mixed models. So what did Chris find? So firstly, for his results, here I'm showing you 2 graphs looking at the proportion of containers where both species co-occurred in early, middle, and late season. We've got the trash containers with the filled-in symbols, the functional containers with open symbols. What he found was that co-occurrence was common in 2 out of 3 trash and 2 out of 3 functional container types. Right? So co-occurrence was common. Towards the end of the season, about 40% of the containers with mosquitoes had both species in them. So competition is likely to be occurring. And he also found that the proportion of total larvae that were pipiens decreased over the course of the season, which we might expect through larval competition, but he saw milder decreases of Culex pipiens from early to late season in 2 out of the 3 functional container types, which may suggest there's moderate evidence that the competitive effects of Aedes albopictus may be less in these functional containers. What Chris found with his competition trial was a much more clear-cut result. And in particular, he found this interesting container type treatment interaction, which suggests that the competitive effects of Aedes albopictus and Culex pipiens varies between container types. Right? We looked at survival of Culex pipiens and development time of Culex pipiens. And we can see that here in these graphs. Here we've got survivorship, and this should be development time. No, survivorship. Yeah, just looking at survivorship here. What he found was that there was lower survivorship and increased versus decreased treatments for trash container types. Okay, so we should see decreasing survivorship with increasing Aedes albopictus density. Okay, but we see lower survival and increased versus decreased treatments for trash container types. And when we look at mean differences across containers within each type, we see strong effects of Aedes albopictus competition in trash containers, whereas we don't see an effect of Aedes albopictus competition in functional containers. When we have a look at development time, which is measured just by the mean instar that was reached by Culex pipiens after 6 days, we see the same trend. Again, when we look at mean values across containers within each category, we don't see an effect of Aedes albopictus on the development of Culex pipiens and functional containers, but we do see an effect in trash containers. So what Chris really concluded was that overall negative competitive impacts of albopictus on pipiens were apparent at densities relevant to urban container conditions. but there was little evidence of competitive impacts in functional containers. Right? And this is likely due to greater fine particulate organic matter, nutrients, and microbial resources. So this was really the first robust study to show persistence of pipiens after Albopictus invasion in urban container conditions. And interspecific competition is likely important in structuring these communities. Right? And this has implications for disease and control. So persistence of pipiens with the Albopictus may increase transmission risk. Right? Persistence of pipiens, the principal vector of West Nile, which amplifies it among bird populations, is likely to keep West Nile risk elevated. And then with its coexistence with Avius albopictus, that may mean that we have a more efficient bridge vector into human populations. We also find that functional trash cans are particularly good habitat for Culex pipiens. If you looked at those mean differences between trash and functional containers, a lot of it is driven by functional trash cans. Right? They just looked particularly good habitat. And so control really requires resident-based behavior change. We really require residents to put the lids on their trash cans, which is a simple behavior, but routinely it's not done. So with that, I just want to conclude. I've presented a couple of studies which has shown how socioeconomic factors can influence mosquito ecology at different scales. Cities are heterogeneous socio-ecological systems. So social factors can play an important role. And really our capacity to detect and control disease is really impaired where the lack of data on vector biology at within city spatial scales. And when you look at the literature, we don't have really good data on within city scales, right? Between neighborhoods that vary in social and ecological context. And I think that's probably a focus of research as we move forward, especially in temperate cities where the risk of emerging diseases like Zika and chikungunya may be lower than cities in the southern continental United States, but it's still an important risk that we should pay attention to. Okay, with that, thanks very much for listening.
Thanks, Paul. We do have some time for questions, so I would encourage people from the audience, and then we'll be watching online. So go ahead and just ask the questions. Yeah.
One of the key distinctions that you showed with the aerial map between the low and high income neighborhoods was vegetative structure.
Right.
And you looked at that as a factor. You know, I'm thinking in terms of bird presence,
Right.
and also even the male mosquitoes and the habitats they might be utilizing.
Yeah, so the Baltimore ecosystem study.
Can you repeat the question?
The question is that different neighborhoods, or neighborhoods vary in their vegetation structure. That this could affect mosquito abundances, and it could affect bird abundances, potential blood meal hosts. And so in response to that, the Baltimore ecosystem study has done mist netting among neighborhoods in Baltimore and shown bird populations do vary. We've got blood meal analyses which do suggest that mosquitoes take a higher proportion of bird blood meals in more higher income neighborhoods and potentially bite different mammalian hosts in low income neighborhoods like rats and dogs. In regards to directly affecting mosquito abundances, I think that's a really interesting question. Because especially vacant lots, which vary quite considerably in vegetation type and amount, could provide different resting sites and adult feeding opportunities in occupied lots. And so actually we have a PhD student the next summer is going to be vacuuming mosquitoes in different lot types to see if adult populations vary between parcels of land or lots that have radically different vegetation types. Yeah.
Your studies on the competition are very interesting. Do you know the mechanism, like how these larvae are competing with each other? Is it due to resources available?
Yeah, so the question was that what is the mechanism by which the larvae compete with each other in these container habitats? Yeah, so the principal mechanism is resource competition. Right? So these container habitats are resource limited. Typically detritus falls into these containers, and the mosquitoes browse on the detritus and the associated microbes. And we know through behavior studies that Aedes albopictus forages much more often than other species, and they filter feed and they browse, which makes them more susceptible to predation. But they're a particularly good forager, and they are likely to convert that microbial resource into biomass much more quickly than Culex. And saying that, interference competition may also play a role. Mosquitoes excrete ammonia, and so some studies have suggested that mosquitoes vary in their tolerance to ammonia. So interference competition may play a minor role.
So what happens in the trash cans, for example? Like, how does it dampen that competition?
So essentially, you're just raising resource levels to a really high level, and you're relaxing competition. Resources are in abundance. And when you look in the trash can, it's really clear that resources are pretty abundant. Yeah.
One of your slides said 0.33 larvae per milliliter. So that's 300 larvae per liter.
Yeah, so that was a really high baseline density. So that average includes only containers with mosquitoes. And it can include containers with really high Culex levels. Culex lays egg rafts, and you just get super high densities of young install larvae. Yeah, it's really high.
How many adult mosquitoes would you get off? What's the ratio?
Right. So that's a difficult question to answer. That can vary depending on resource levels typically. But I think one of the photos that I've shown is a recycling bin with a lot of detritus in it. And I would suspect that could produce thousands of mosquitoes within a very short time. If you think of one egg raft of Culex, it could be 100, 150 young larvae, and if they're not suppressed by limited resources, then potentially a large proportion of those could emerge into
Could you just repeat the question from the audience? They can't hear the question, so if we could just keep
Sure.
Yeah.
Yeah, Herb.
Paul, two quick questions. Is there any evidence that any of the neighborhoods are hiring pest control companies to do adult mosquito control in their yards? And two, does the storm sewer system play any role in habitat for either of these species in Baltimore?
Yeah, so the question was in two parts. The first one was that is there any evidence that residents are hiring private pest control to do mosquito control, and what is the potential role of sewer systems in mosquito production? So the first response is that there's very limited evidence that people in low and medium income neighborhoods are using private firms to do pest control. And even in high income neighborhoods, typically, from what we've assessed, people do not. The second response is that we have surveyed mosquitoes, lifted manhole covers, and looked for mosquitoes. And in our study sites, we don't see catch basins which hold water. we're seeing the water continually running. And so we've seen very little mosquito production in subterranean habitats, at least in our area. Now in Washington, D.C., we've done a similar thing, where they do seem to have a catch basin. And that does produce both Culex pipiens and Aedes albicans.
So yeah, I found it interesting. You correlated, what you're finding, mosquito populations to economic levels, high, medium, and low. Have you thought of correlating it to other factors? I know you showed the one about the rain. There was an issue with, inadequate moisture. I mean, have you related it to any other of these factors as well?
Yeah. So the question is that we've related mosquito abundances to socioeconomic or income levels, and have we thought about relating it to other factors? And the response to that is yes. So we've got Shannon's looking at remote sensing data, and we're relating vegetation type to adult abundance data, which kind of also gets at the question of whether or not lots that vary in vegetation perhaps have different adult abundances. And there's lots of dimensions to socioeconomic status. Income's one of them. Vacant lots is another. We're relating mosquito abundances to education outcomes, to certain other interventions, to weather patterns, rainfall. I think they're really the main suite of other factors that relate to mosquitoes.
Thank you, Paul, for your expertise. You've related the prevalence of mosquitoes to vacant lots. Is there any inference you could make between prevalence of mosquitoes to community gardens that are now, being put into the vacant lots?
Right, so I think the question is of related mosquitoes to vacant lots. Are we seeing any effect of interventions?
Community gardens.
Community.
We have community gardens being erected into the vacant lots. Vacant lots being used for gardening.
Oh, gardening. Right. Right. Yeah. So, is the role of community gardening affecting mosquito population? So, within our study sites, we have got a couple of community gardens, and from memory, I think mosquito abundances may be slightly lower in community garden plots. I think the Baltimore ecosystem study has got a range of interventions, and Baltimore City has got a range of, treatments to vacant lots. Interestingly, a lot of vacant lots, though, still have structures on them, which we can't access. Right? They're boarded up. It's illegal to go into these vacant buildings. We suspect that those vacant buildings may be a good source for mosquitoes, even if we do our best to remove containers from the area outside the building. Related to that, we did an intervention just this past summer where we did an intensive trash cleanup on some replicated blocks. And we compared mosquito abundances before and after the trash cleanup on these blocks and control blocks that didn't do any cleanup. And we didn't see any change in mosquito abundances. And we suspect that could be because we're just not able to access a lot of trash containers in vacant buildings. It was very disheartening. We spent a lot of time, we were carrying out used tires, we were getting in there, we had an army of people. There's a lot of trash, and we did our best over a couple of days to get rid of most of it. And we didn't see any change in mosquito adult abundance.
Did you bring it up to the level of the high income, where you said those? I guess I wonder, when you did remove it, did you take it to the level of what you would see in a high income, or it was just removal? Okay.
So we had control blocks, so we did no removal. We had intervention blocks where we did an intensive removal. We tracked before and after. And these were blocks with a lot of vacant lots, where we thought we could really make a difference. And we didn't. And we've seen in New Jersey some results consistent with their outcome, and some results that have seen a measurable decline with really intensive trash removal. But I suspect it really requires residents, to be removing trash, to be on top of things week by week, and the vacant lots is a real issue.
One more question. It's hand over there. Glenn?
That's for two, but I guess I only get one. You mentioned early on about the difference between larval control versus adulticiding. So what are your thoughts about effective response in terms of managing disease risk? I mean, should we go back to adulticiding, or does that have too many other problems? Or?
So the question is that should we go back to adulticiding to try and combat disease risk, especially I guess with these emerging pathogens? So that's a really good question, and I'm certainly no mosquito control expert. I feel as though I'm not an expert in mosquito control. And we do see that the response down in Florida has been to aerial spray some areas with adulticide, with organophosphates, right? And it does suppress adult populations substantially. So I suspect if there is an outbreak, that is the most effective way to control mosquitoes. And there may be situations where that's called for. But of course, there's non-target impacts. And I guess I don't know the good evidence, but honeybees may be susceptible to some of these insecticides, and you can't control where these insecticides are going. I think that's a good question. I would suspect maybe in certain situations it may be called for. But I think long term we should certainly be thinking about these other much more sustainable options. Source reduction, these biorational larvicides would be the long-term options.
Well, thank you, Paul, for your very informative talk, and look forward to speaking with you later. But we're going to pause at this point for break. So you folks online, we'll be back at 10:20 with our next presentation. So thanks again.
Thank you.